Hello students, today we will see the next topic in the chapter protein. In the previous lecture we have seen the introduction, what is the classification of the protein, what are the importance of the protein, and one of the method, that is Jadhav method, we have already seen. Today we will see different methods for the analysis of proteins. First one is the Dumas method, then second one is the IR spectroscopic method, and third one is the Biot method. Let us see one by one. So first one is the Dumas method. So the combustion method was introduced in 1831 by Dumas. It has been modified and automated to improve the accuracy since that time. Samples are combusted at high temperature around 700 to 1000 degrees Celsius with a flow of pure oxygen. All carbons in the sample is converted to carbon dioxide during the flash combustion. So the nitrogen containing components produced include nitrogen element, molecular nitrogen and nitrogen oxide. So the nitrogen oxides are reduced to nitrogen in the presence of copper under a reduction column at a higher temperature about 600 degree Celsius. So the total nitrogen, which is consisting of the inorganic fraction, that is comes from the nitrate and nitrite, release is carried by pure helium, and it is quantitated by gas chromatography using thermal conductivity detector. So the ultra high purity acetanilide and EDTA, that is ethylene diamine tetraacetate, may be used as a standards for the calibration of nitrogen analyzer. So the nitrogen determined is converted to protein content in the sample using protein conversion factor, and in this way we have to calculate the amount of proteins present in the given sample in terms of nitrogen. Now look at the procedure. So samples generally we have take about 100 to 500 mg of the sample, and they are weighed into the tin capsule and introduced to the combustion reactor in automated equipment. So the nitrogen released is measured by built-in gas chromatograph. Okay, so this is the instruments or equipment. Here we have to put the sample into the combustion zone at higher temperature about 700 to 1000 degrees Celsius. Then after the combustion, the material is sent to the reduction compartment. In this compartment there is a reduction of nitrate and nitrite into the nitrogen is going to take place, and then that response is the particular components are sent to the GC, and from that GC the separation is occurred, and that separation is detected by using the thermal conductivity detector. Now what are the applications? So the combustion method is an alternative to the Jadhav method, and it is suitable for all types of food. As we have seen that in case of Jadhav method, we have find out only the proteinaceous nitrogen. Okay, but here we have used, this is for all kinds of foods, and especially this method is useful for analysis of meat and cereal grains. Then what are the advantages? So this particular Dumas method, it requires no any hazardous chemicals for the analysis. It also require very less time, around it should be completed within the 3 minutes. Then recent automated instruments can be analyzed up to 150 samples without any kind of attention. So this is the automation in the instrumentation, that's why number of sample we have to analyze, it is somewhat more. Now what are the disadvantages? So here we require the expensive equipment. For example, if you consider the GC column, it is quite expensive. Also combustion equipments are expensive. Detector is also expensive, that's why this instrument is quite expensive. Then it measures total organic nitrogen but not a protein nitrogen. So it just only determines the total organic nitrogen. Means what? The nitrogen from other sources, also from vitamins or any kind of source, it can be detected. And our motto is that we have to find out the nitrogen present in the protein. So for this is one of the disadvantage of this method. Now the next one is the IR spectroscopy method. So the infrared spectroscopy measures the absorption of radiation in the near or mid IR regions by the molecules in food or other substances. So there are different functional groups in the food which absorbs different frequencies of radiation. For the proteins and peptides, various mid-IR bands and near-IR bands are occurred, such as 3300 to 3500 nanometer, 2080 to 2220 nanometer, then 1560 and 1670 nanometer. These are the characteristics of the peptide bond that can be used to estimate the protein content of the food. By irradiating the sample with wavelength of infrared light specific for the constituent to be measured, it is possible to predict the concentration of that constituent by measuring the energy that is reflected or transmitted by the sample. So that energy is again inversely proportional to the energy to be absorbed. Now, what is the procedure? So, IR spectroscopic instrument, it is a very sophisticated instrument. In this instrument, we just have to put the sample on the suitable accessory. So, depending upon the nature of the accessory, we have to put the sample on the accessory. We have to take a spectrum from the proper software and then interpret the spectrum. Here, if you have the diffuse reflectance accessory, then we have to put the particular background, put the KBR for the background, then we have to mix the sample with the particular KBR and then put the and take the spectrum. While if the attenuated total reflection accessory is there, here the total reflection of the IR radiation is there, and that's why there is no need of any kind of standard in the analysis. So these are the this is the procedure for the analyzing the sample by using IR spectroscopy. Then what are the applications? So the mid-IR spectroscopy is used in the infrared milk analyzers to determine the milk protein content, while the near-IR spectroscopy is applicable to the wide range of food products, for example, grains, cereal, meat and dairy products. Then this particular instrument, it is quite expensive and they must be calibrated within the proper duration. However, the samples can be analyzed very rapidly. Around 30 seconds to 2 minutes are required for the analysis of one sample by the analyst with very less training. There is no such kind of training is required for the analyzing a sample. Now the next one is the biorate method. So the biorate method, it is the method in which a violet purple color is produced when cupric ions are complex with the peptide bonds. So the substances containing at least two peptide bonds. If you see this particular figure, in that case we will have see that the copper forms a chelate, or we can say the complex with the peptide nitrogens under alkaline conditions. So whenever it forms a complex with that particular copper, then it shows some kind of color. So the absorbance of the color, which is read at 540 nanometer, and the color intensity, and as we know that the color intensity, it is directly proportional to the protein content, or we can say the concentration of the sample. Now procedure. So for that purpose, we have to first take 5 ml of the biorate reagent in a reagent which is mixed in 1 ml portion of the protein solution. So that protein solution containing around 1 to 10 mg per ml of the concentration. Then the reagent includes copper sulfate, NaOH and sodium potassium tartrate, which is used to stabilize the cupric ion in the alkaline solution. After then we have to mix the reaction mixture well and then allow it to stand for room temperature for 15 to 30 minutes. Then there is a color is developed to the solution. Then the absorbance is measured at 540 nanometer against reagent blank. Now Filtration or centrifugation before reading absorbance is required if there is any kind of precipitation or any kind of small particles are there, then we have to filter the given solution. Then after getting a particular absorbance, we have to first plot a standard curve. So for plotting a standard curve in case of any kind of protein sample, we have to generally use the bovine serum albumin as a standard. This is because by using this solvent or by using this standard, we can get an exact reading of the proteins. So here why BSA is used? Because it is cheaply available and it is a universally used protein standard for the various kinds of estimation of protein samples. Then what are the applications? So first one, what are the applications? So in that, the Biret method has been used to determine the proteins in the cereal, meat, soybean proteins and quantitative tests for the various kinds of animal feeds. The Biret method is also used to measure the protein content of isolated proteins. Now what are the advantages? So the first, it is less expensive than Jadhav method and it can be very rapid. So we have to require around 30 minutes to complete the analysis and it is the simplest method for the analysis of the protein. Then the color deviation are encountered less frequently than the Lorry method, ultraviolet absorption or turbidimetric method we have already seen in the next lecture. So very few substances other than proteins in the foods interfere with the Biret reaction and it does not detect the nitrogen from the non-peptide or non-protein sources. So this is one of the advantage that we have to selectively determine the nitrogen content which is originated from protein. If there is any kind of non-protein sources there, it cannot be detected by using Biret method. So this is one of the advantage while considering the Biret method with the others. Now what are the disadvantages? So this method is not very sensitive as compared to the Lorry method and it requires about 2 to 4 mg protein for the assay. So the absorbance could be contributed from bile pigments if present. So generally what happens, if some kind of food sample or animal sample or whatever samples are there, it contains some kind of bile pigments. If bile pigment is there, it contributes into the absorbance of the given solution and hence we cannot get a proper analysis. Then high concentration of ammonium salts interfere with the reaction, as we know that if there is ammonium ions are there or salts are there, they will involve in the particular complex formation in the particular peptide and hence we cannot get a proper analysis results. So generally what happens in this case, color varies with different proteins. Suppose if you consider tryptophan, tryptamine, lysine, so these are the having different kind of nature and hence they are showing different kind of color and that's why we cannot effectively analyze the proteins. So gelatin gives a pinkish purple color. So there is some kind of opalescence is occurred whenever there is other entities are present in the given sample. So this is one of the disadvantage of Biret method. So this is all about these three methods. Now in the next lecture we will see about the Lorry method and dye binding methods. Thank you.